We will start now with the second plenary talk, and we are particularly happy to introduce Olivier Faugeras as second main speaker. As you all know, he has initiated this series of ICMNS conferences in 2015 in Saint-Jouan-les-Pins. Olivier is a member of the French Academy of Science. He is a research director emeritus at INRIA in Sofia Antipolis, where during a long time he was the director of a research group called Neuromatcom, and that's a project that he actually initiated. He is, I would say, one of the leading figures of the mathematical neuroscience community, and actually he did a lot for this community. For example, with the creation of the two journals, Journal of Mathematical Neuroscience, and then the follow-up, the Mathematical Neuroscience and Applications. With his main scientific contributions, Olivier has built a bridge between the deterministic world, so the world using dynamical systems theory, bifurcation analysis, and the stochastic one, using stochastic processes mainly by the means of large deviations. He is interested in a lot of things, among others in the asymptotic properties of large systems of interacting neurons, the rigorous proofs of the mean field limits, in the theory of neural fields, and the applications to the modeling of visual perceptions. He is also interested in plasticity, in learning. He also worked on random synaptic weights, Hopfield neurons, and so many other things that I'm not going to talk about all these. Olivier received for his scientific work several awards and prizes, in particular the Asriel Rosenfeld Award for his work in computer vision. And today he will speak about neural fields, from vision to consciousness, a prospective mathematical perspective. And so we are very curious about what you're going to tell us, Olivier.
Thank you very much, Eva, for this kind introduction. And thank you to all of the committee for inviting me to this conference. So you said the title, I won't repeat it. This is an outline of my talk. I'll start with a short introduction. Then I'll discuss the mathematical study of the neural field equations. Then I'll see some applications, including symmetries to visual orientation. I hope to have the time to talk about stochastic neural fields, and then I'll dive into the question of consciousness. I haven't worked on consciousness, but I think it's something some people in our community should work on, and I will conclude. So the introduction. Mathematical neuroscience is really, the goal is really to develop predictive and possibly falsifiable, a la Karl Popper, this British philosopher, theories. One of the most famous ones, one of the most famous theories, such theories, is found in the work of Hugh Wilson and Jack Cowan, published in the years 72 and 73. Here are pictures of Hugh and Jack and their papers, the landmark papers that they published in Biophysical Journal in 1972 on neural masses and on neural fields in 1973. Let me flash the celebrated Wilson and Cowan equations at you. They are written here on the slide. If E of t and I of t are the average membrane potential of excitatory and inhibitory cells at time t, then you have those two nonlinear differential equations. I just comment a little bit. The first one, tau d dt equals minus e. That's just an exponential decay toward the initial condition. And then you have a linear combination with weights j sub e, j sub I, e of nonlinear functions of e and I. Se and Si are the usual sigmoids. We'll hear a lot about sigmoids later in the talk. And IE X is the external input to the excitatory neural population. So this is the voltage-based neural mass model. There are also activity-based neural mass models. So this is this is what a neural mass is, and we'll see neural fields a little bit later. So after this very short, brief introduction, I will talk about the mathematical study of the neural field equations. It starts with the work of Wilson and Cowan again in 1973, and followed by Amari, a Japanese researcher, in 1977. And it was followed by many attempts by theoreticians and mathematicians who tried to characterize the solutions. There's a long list here. Breslov, Coombs, Amontrot, and many others, which I won't have time to read. Today, I think it's accepted in the mathematical neuroscience community that the correct framework for analyzing these equations is functional analysis, because you can use such functional spaces as Banach, Hilbert, Sobolev, which are quite handy. You can use fixed point theorems to prove existence and uniqueness of solutions. You can use spectral theory to analyze stability. You can use bifurcation theory in order to analyze how the solution changes when you change parameters. Let me write them for you, show them to you. They resemble the previous equation, the Wilson and Cowan equations, with some change in notations. So I'll read it. V dot of x and t equals minus v of x and t plus j of t applied to s of lambda v at x plus some external current. And there is an initial condition v at time 0 is equal to v naught. So what are these quantities? V and v naught are p-dimensional vector functions, which are defined on a set omega, which can be an open, bounded, or unbounded piece of R2 or R3, or a more complicated space. And they represent average membrane potentials of the neural field, like in the Wilson-Cowan. They both belong to some functional space F. Now, J of t, this is the operator notation I have here. J of t is a linear operator which maps the functional space F to itself. It's also called the connectivity kernel. And it describes the intensity of the projections from the masses at x prime to the masses at x. And this is the linear superposition integral, which is shown at the bottom of the slide. Now, let me comment a little bit about the other quantities that appear in the equation. S is a sigmoidal function, which is basically regular and bounded, like in the Wilson and Cowan equations. Lambda determines the stiffness of the sigmoid, the important parameter. I will use it as a bifurcation parameter later. And x, which lives in omega, represents space or features or more complicated things. Now we have to decide on F. So there are three criteria that we can follow for choosing F. The problem should be well posed. The equations should be solvable. We should also worry about the biological relevance of having membrane potentials functions living in F, and it should also allow numerical computations. So one of the usual choices is the Hilbert space F equals to the set of square integrable functions values in RP and defined on omega. You can also, if you want, impose more spatial regularity, but I won't need it in this talk. J, the connectivity kernel, also lives in an L2 square, a set of square integrable functions. And if it lives in that set of square integrable functions, then J of t defines a continuous linear operator from F to F. So the superposition integral is well defined as a mapping from F to F. Now I rewrite the neural field equations as an ODE, ordinary differential equation, which is defined on the functional space F as dv dt equals minus v plus r of tv. And initial condition is just a function v naught in F. And r of tv is the sum of the operator j of t applied to s of lambda v plus the external current. And in a 2008 paper, SCIEM paper, together with François Grimbert and Jean-Jacques Slotine, we proved that if J is a nice mapping, continuous mapping from R plus the time to L square, and its F norm is bounded for all T's, And if Ix, the external current, is also a smooth continuous mapping from time to functional space, then for all initial conditions, there is a unique solution V defined on R plus for all time and continuously differentiable of the Cauchy problem. Sorry. I would like to mention briefly here some closely related work by Roland Pothast and Peter Baumgraben, published in 2009 in the Mathematical Methods in the Applied Sciences. Now, we have existence and a uniqueness in this case. What about boundedness and stability? Well, we also had these results with François Grimbert and Romain Vels in neural computation. If the external current is bounded in F norm, then the solution is also bounded. Your membrane potential does not explode, doesn't blow up. And furthermore, if the product of the stiffness by the spectral radius of the symmetric part of the operator J is less than 1, then every solution is globally asymptotically stable, and in particular, it is unique. Okay, so we would like to learn a little bit more about these solutions. And in particular, I would like to state a few results about persistent states. They are important because persistent states or stationary solutions that do not depend upon time are pretty good models of memory. So I will have to assume that the external current and the connectivity function do not depend upon time. And I will rewrite the neural field equation, no more time derivative, as f of v and lambda. Lambda, remember, is the stiffness of the sigmoid. So two main questions we tried to answer in the past. How many solutions can there be? We saw that under some conditions there was only one. And what is the manifold of solutions when you vary the parameters? So we have this important proposition in a paper with Romain Vels in SIAM in 2010. If I define the set of solutions for a given value of lambda as such here, b lambda as the set of solutions, which I note vf lambda, then first of all, we proved that the norm of the difference with the solutions for lambda equals 0, so it's a flat solution, is bounded by a function, upper bounded by a function of lambda, which is also bounded, which means that the solutions live in the white part of the right-hand side picture, which shows the horizontal axis is lambda and the vertical axis is this infinite dimensional space where V lives. And of course, we just represent the norm of V. So they are bounded in that space. And the second thing we proved is that if the number of solutions is finite, then this number must be odd, as shown in the picture, 1, 2, 3. And we proved this by using the Loray-Schauder degree theory. The third result we had was that if you consider two values A and B of lambda shown on the right-hand side picture, and if I consider the sets B lambda times lambda, so these are the vertical lines and points on these lines corresponding to the solutions, then there's always a connected component which intersects B sub A and B sub B. So the left-hand side of the picture is impossible. There's no path, but the right-hand side is what happens. The green path connects B sub A to B sub B. And we did that. We proved that using the Loray-Schauder theorem. So we have pretty good global information about how about the geometry of the stationary solutions. That was in that paper. Okay, so that's the end of the mathematical study of the neural field equations. I want to apply this to visual orientation sensitivity, or how symmetries make things more complicated. So we know from neurophysiology that visual area V1, shown in red in the slide, is organized as a set of orientation sensitive columns and hypercolumns. This is the beautiful work of David Hubel and Thorsten Wiesel in 1974. And this is shown in the right-hand side, the right picture. Visual orientations in the retina are coded in color. So color is a visual orientation. And if I plug in an electrode here at this point on the cortex, this is a visual cortex volume, this is the surface here, I notice that the neurons respond only to the red orientation. Okay, and if I go deep in the cortex, this sensitivity doesn't change. But if I move about, then this sensitivity will smoothly change, moving from red to purple to green. And along these white curves, it's pretty smooth. It's constant orientation. And you can see that these curves meet at some points, which are singular points. And these points are pinholes, called pinholes, and will play a very important role in what follows. Okay. If I look at the cortex from above, I see this map of colors, and this is called the preferred orientation, or PO map, and it's periodic because of the pinwheels. This periodicity will turn out to be important. Now, the neural field model I can write for V1 is very simple. It's a classical one, as before, dv dt v equals minus v plus the superposition integral. I changed the lambda to sigma to be compatible with the figures I had at my disposal. And now we will focus on the connectivity function J, which I will assume to be the sum of two parts, a local part, J loc, which is a translation invariant. I forgot to say that X and Y are points on the surface of the cortex, so it's translation invariant, plus epsilon, where epsilon is a small number, positive number, much smaller than 1, a long-range connectivity function, J sub LR. Okay, so J loc, the local connectivity function, is a Mexican hat-like, so it has local excitation and local remote inhibition, plus and minus. Now, the bifurcation analysis of the homogeneous model, i.e. the model when epsilon equals 0, is very well understood. We solve in the square omega with periodic boundary conditions, therefore omega is actually a torus. And we know that there is the emergence of Turing patterns as shown on this bifurcation diagram. The horizontal axis, again, is the stiffness sigma of the sigmoid. The vertical axis is the infinite dimensional solution. And that is well known, for example, see the paper by Coombs, Baumgraben, and Podhast in the book they recently edited in 2014. We know that there's an emergence of Turing patterns. There are stripes, as shown here, and I have a problem with my mouse, stripes and spots. So stripes are always unstable, spots are stable on a very short interval, and then you have two subcritical secondary branches that recover stability. Stable is shown in orange or yellow if you on your screen maybe. Okay. So this was with the symmetries of the local orientation, local connectivity function. Now, I said before that because of the translation of invariance, the solution lives on the torus. Now, I will assume, I haven't talked about the PO map, but I will assume that it is periodic, so it's approximately periodic, and the period is the same as that of the periodic solutions. This is fairly well supported by biology, paper by Kenneth and colleagues. So what I'm showing here is the PO map. Again, remember that orientations are coded in color. The black lines surround the hypercolumns, where at the center of the black squares you have a pinhole, and the white lines correspond to the square, the fundamental domain. So the pinwheels are shown either in black or white. And notice that if I rotate, if I rotate about a pinwheel, the color will change in different ways. In other words, black pinwheels correspond to a clockwise changes in color, whereas the white one counterclockwise. So pinwheels are clockwise or counterclockwise, meaning that if I rotate about, then colors change differently. Now, let's talk about the long-range connections. The long-range connections will make things very interesting. J sub r is given by this interesting expression, which I will not comment accurately, but I will use the biology here. Again, we'll look at the PO map, and there's a center here, which is sensitive to purple color. So this is one orientation. Then through the injection of some dye, by tracing how the injected dye progresses, we can tell that these particular neural masses are connected mostly to other neural masses remotely, which are sensitive to pretty much the same orientation. Okay, so it's patchy and it's orientation sensitive. This is a diagram which is taken from the landmark paper by Breslov et al. In 2001, which shows that if I point X, I'm here, I have various columns sensitive to various orientations. And you see that this one at X is only connected to this one at Y with the same orientation. And the same here, this one has a different orientation sensitivity, and it's only connected to the one at Y, which have similar orientations. This is very, very important, and this is gathered in this formula. Now, when you study the bifurcations or the changes in the solution, and there is a group underlying your model, it's important to look at the symmetries of the PO map of your equations. So the most important symmetry here is the action of the rotations with center pinwheel on the PO map. So if I rotate a point X with center P of an angle phi naught of pi over 2, what is the value of the PO map at this new point? Well, it's very simple. You can check it by yourself, equal to the value of the PO map at X plus epsilon phi 0 by 2, and that is epsilon pi over 4. And epsilon is 1 if P is counterclockwise. Remember that the pinwheels are counterclockwise or clockwise, and it's -1 otherwise. Okay, so if we want to study the changes in the solutions, we form a set omega with 2n+1 copies of omega naught. Remember the white square. The symmetries of that set are the symmetries of the square, D4, semi-direct product Z modulo 2nz square, since we are in 2 dimensions. So that's the symmetries without the PO map. And it turns out, very interestingly, that the PO map reduces the symmetry to one of the 17 wallpaper groups or crystallography groups, which have been studied by mathematicians since the 19th century. And further constraints from the PO map reduces this whole set of groups to just 2, called PMM or CMM. And they are shown actually, they are shown here and here together with the PO map. Okay, so we have this extra group of symmetry that acts on the PO map. And what happens? Well, what happens is that we're going to lose some symmetries in these solutions when we turn the long-range connectivity on when epsilon is non-zero. So as shown in our paper, in 2015 paper in the JMN, by the way, the symmetry group gamma of the NF equation, neural field equation, is one of these crystallographic groups, P4M or P4, meaning what? Meaning that if I call f of v of lambda the neural field equation, and if I call little gamma an element of this group gamma, which is equal to P4M, then if I apply gamma to f of v and lambda, it's just the same. It's equal to f of gamma applied to v. So that means mathematically that the neural field equation is equivalent with respect to the group gamma. So it's well known when you turn on the long-range connections, the system undergoes symmetry breaking. The solutions will preserve only the symmetries of gamma or some subgroups of gamma. Okay, so we understand the symmetries. What about the dynamics? Well, the dynamics can also be studied. I will try to explain this briefly. Remember that the solutions live on a torus when there is no long-range connection. So I call T naught the unperturbed torus, which is V0 of X, and X is in omega. And we add, when you add the long-range connection, then there you have a torus, flow invariant torus TLR, which will persist as long as the perturbations are small. Epsilon is very small with respect to 1, small enough. So this was shown, for example, in the beautiful book by Pascal Chaussat and Rainer Lauterbach, Methods in Equivariant Bifurcations and Dynamical Systems in 2000. So questions we can ask. So how many steady states do actually persist? And what are the observed phase portraits? Well, the topology of the torus is an important constraint. As shown here, we have this proposition in the same paper. If we assume that there is a finite number of equilibria on the perturbed torus, which are all hyperbolic, non-degenerate, non-zero real parts, and if epsilon is non-zero and the anisotropy is real, that is key, the parameter key, then there are at least 8 equilibria on the perturbed torus, 4 of which are saddles and the other 4 are nodes or foci. And the proof is a simple application of the Poincaré-Hopf theorem that tells us that if n is the total number of equilibria, that is the sum of the number of foci and the sum of saddles, n minus 2s is equal to chi, the characteristic of the torus, which is equal to 0, and hence f is equal to s. So then if we look at rotations, the rotations with respect to the pinwheels, we find that the number of foci is equal to 4. Therefore, we have 4 saddles. This is a very deep and very interesting theorem proposition. And actually, you can verify it numerically. You can draw phase diagrams like here, where you can, this is a simulation in omega. You have these 4 saddles here, and then you have 1/2 foci here, 1/2 foci here and here, that's 2 foci, and 1/4 of a foci at the 4 corners, that's another foci, 1 at the center, that's 4 foci and 4 saddles. Ooh. We cannot, so there's dynamics, dynamics, I forgot to say, there's motion in here. Examples of cortical activations. These are simulations of the equilibria. We superimpose the activity map, the membrane potentials, or S of the membrane potentials, to the PO map, and the PO map is semi-transparent. When the activity is very large, the PO map is transparent. The activity map is transparent, so you see the PO map, and you can represent this as a perception, if you wish, by averaging the PO map, with weighted by the activity. So here we see basically patches. Here also we see patches at different solutions, and here we see edges, only one orientation. So here we see all orientations, and here none. Okay, final remarks on this part. It's a pretty wide generalization of the early work of Armand and Cowan in Biological Cybernetics 79 and the landmark paper by Breslov et al. In 2001. And I would like also to stress the fact that, to me, group theory is really underutilized in neuroscience, and we should make more use of it. Okay. Taking noise into account, stochastic neural fields. Okay, so we know that neurons are intrinsically noisy because of ion channels, thermal noise, synaptic noise, and so on and so forth. So we would like to rewrite the deterministic noisy neural field equations a la stochastic differential equation, which I did here. dV equals the drift term plus the diffusion term. S and sigma are globally Lipschitz. S is bounded. Okay, and what is W of T and X? It's a stochastic process in 2 variables. T, it's time, and X belongs to Rn, which is our set omega here. So we have to define what do we mean by a solution, and it has to involve an object, this kind of integral, an object of that form. And I will try to explain this a little bit. Let me also mention that this approach has been used empirically, i.e. without worrying about being mathematically correct, by Breslov and Weber, Breslov and Wilkinson, Kilpatrick and Ermantraut. So one of the approaches is to use the theory of Walsh stochastic integral, published in 1986, which allows you to cleanly define a white noise W dot on R plus times Rn, and then a white noise process Wt of A, where A is a Borelian of Rn, and a set of functions Pw that can be integrated against the white noise process W, which is exactly what we need to make sense of this object here. Okay, so Walsh has done the work. There are some problems. Informally, you can think of the object W dot as a random distribution, very rough. So any solution of the stochastic neural field equation is distribution valued in the X direction, which is the space direction, and that's no good. So you can smooth that out by defining a slightly different noise, Gaussian random field, by basically convolving it with a function in L2. This is the integral here. And it's easy to verify that phi of x minus dot is in the set of integral functions against W. You can prove that W phi is regular in time, same as the Brownian path, i.e. Hölder continuous for alpha strictly less than 1/2. And Hölder continuity in space, which is also nice, can be enforced by imposing a very weak regularity in phi, which is indicated here. Tau sub z of phi is just translated by z. Phi is just a function. If you take the L2 norm of the difference, then it should be less than c phi, the Euclidean norm of z raised to the power alpha. So if you do that, if you have this condition C1 for all t, the function x goes to W phi of tx, it has an eta Hölder continuous modification for all eta less than alpha. That's fine. That's nice. Now we can rewrite using this W phi, we can rewrite correctly, as I will show, the stochastic neural field equation driven by spatially smooth space-time white noise as shown here. And by a solution, we mean a real valued random field as shown here. So you have the deterministic integral here and the stochastic integral there. So together with James Inglis in 2015, we did the obvious thing. We looked at the sufficient conditions on the connectivity functions for the existence and the regularity of the solution. So we proved this theorem of existence and uniqueness if for all x in Rn, the function y goes to j of x and y is in L1, sorry, and the sup with respect to x of its L1 norm is bounded, then you have existence and uniqueness. I won't go into the details of stated in the theorem. And it's a fairly straightforward application of basic principles in stochastic calculus. Same with the spatial-temporal regularity of the solution. If you add this C3 condition on J, if you take the L1 norm of the difference for 2 points x1 and x2, so the difference J of x1 dot minus J of x2 dot, and if it's upper bounded by some the norm raised to the power alpha of x1 minus x2, where alpha belongs to 0, 1 semi-open, then you have regularity. You have eta 1, eta 2 Hölder continuous in time and space. So we're happy we have something we can work with. The final remarks, I'm catching up on time. The final remarks are that probability theory, stochastic calculus, and large deviations, I don't have time to talk about, but Eva mentioned it, are very important tools in mathematical neuroscience and should be put to good use. Consciousness, last part. Consciousness, in the late 80s, consciousness was a tolerated hobby for the aging scientist. I guess that's the class of age I belong to. One of the problems is that consciousness, the word consciousness, is loaded with very fuzzy meanings. You can be conscious of something, you can be conscious. So you have to distinguish between at least 3 meanings: wakefulness, vigilance, attention, focusing your mental resources, and the fact of being able to report what you see, what you hear, what you think. So that's called conscious access. And recently, and most obviously in the work of Stanislas Dehaene at the Collège de France, there are three major ingredients that have been made available to researchers to make consciousness amenable to scientific inquiry. So first ingredient is the focus on conscious access, being able to report. The second ingredient is manipulating conscious perception in the lab. And the third ingredient is to carefully record introspection. And of course, thanks to the new quote and unquote brain imaging modalities like functional MRI, EEG, electroencephalography, MEG, magnetoencephalography, or optical imaging, not for humans. So an example of manipulating conscious perception. Here you have a sequence of images. You show either random shapes, which are called masks, or a 33 milliseconds hidden word. We hear the word radio, and post-mask, again, you mask. And for 33 milliseconds, you don't see consciously the word radio, okay? So if you increase 33 milliseconds to 100 milliseconds, say, then radio will become reportable. You've seen it, okay? But your unconscious has seen it. That's important. And then there's a second example below. Now, what is the unconscious and what is the conscious? I will not answer these very deep questions, but I can follow the end in saying that virtually all the brain's regions can participate in both conscious and unconscious thought. For example, I won't go through all examples, but the unconscious meaning, you can understand meaning of a word without being conscious of seeing the word. So for example, if I show you the word red for a very short amount of time, like in the example before, then this word will prime the corresponding color. By this, I mean that if I next show you a number of colors and ask you to choose one of them, then you will choose with a high probability the red one. So the word red has been understood by your unconscious processing system, and it can prime your action. Okay, so what is the unconscious? Some of you may have read the beautiful book by Henri Poincaré, a French mathematician, published in 1902, and he says that the subliminal self, that is the unconscious, is in no way inferior to the conscious self. It is not purely automatic. It is capable of discernment. It has tact, delicacy, and it goes on and on and on. So the unconscious is indeed very, very, very powerful. Then what is consciousness good for? Why do we need consciousness? Well, at least for these four reasons here, it seems that the unconscious is actually doing statistics. So it's keeping, if you have a word like bank, which have two different meanings, the bank money or the bank of the river, the unconscious keeps both meanings ready for use. And the conscious, it's only the conscious part of our mind that does sampling. The brain performs some sort of Bayesian inference. It's a very important item. Then the lasting thoughts. Once a piece of information is conscious, it stays fresh in your mind for as long as you care to attend to it and remember it. So that's important when you want to make decisions, for example. The third item is it's only consciously that you can do, you can become a human Turing machine. You can do arithmetic. The tape of the Turing machine is just a piece of paper. So the consciousness gives you the power, us the power of a sophisticated serial computer. And it's also, of course, a social sharing device, and there are other examples. Now, Dehaene and his co-workers have identified 4 signatures of consciousness. The first signature is a sudden ignition of parietal and prefrontal circuits. So if I show you words which are masked as before, so they can be visible or invisible, when they become visible, when they're conscious, you have a huge surge of activity as shown as this by this curve in the parietal and prefrontal circuits. So this is the invisible words, not much activity. And this is the visible conscious world, you can report it, and there's a huge surge of activity. Signature number one. Signature number two, you have a late slow wave, the P3 wave, which can be detected using EEG. So it uses the same kind of experimental apparatus, and it really corresponds to a huge surge of activity as shown here in the prefrontal cortex. And that resembles very much, a good metaphor for this is a bifurcation in the dynamics of the neural networks underlying consciousness. Third signature, so that you can measure using deep electrodes, like in epilepsy patients, they are implanted, they have deep electrodes implanted, and if they agree, you can use these electrodes to perform some experiments. And again, if you show a masked image and if you increase the delay between the mask and the target image, then it becomes visible to your conscious mind. That implies a sudden and lasting burst of high frequency in the gamma band that is larger than 30 Hertz, a huge and lasting burst of high frequency activity. And the fourth signature is correlation or synchronization. Synchronization of information exchanges across distant brain regions. This example here shows, okay, this is a head of a patient with electrodes, EEG. This face is not recognizable, so there is very little correlation or synchronization between the electrodes, whereas when the face is visible and recognizable, then you have a huge amount of synchronization between a large number of electrodes. So these are four signatures which have been, and they're very robust, and they have been proposed by a number of researchers, including Stanislas Dehaene and his co-workers. So how do we theorize consciousness? Well, the awareness of a piece of information, by definition, means that it has entered a specific storage area, which makes it available to the rest of the brain. We don't know which specific storage area, but it's there. And it's an old idea. Hippolyte Taines, the French philosopher, talked about the theater of consciousness in the 1870s. He says that he wrote that the conscious mind is like a narrow stage, theater stage, that lets us hear only a single actor, but there are lots of invisible actors in the background which are active. It's an idea also that was widely used in AI in the 1973, so a century later, by Raj Reddy and his colleagues in the speech recognition system Hearsay. So processors were using a blackboard, the theater of consciousness, to exchange information. And then, and then the psychologist Bernard Bars, this is the same global workspace, which is indicated here. This is the conscious global space, and you have receiving unconscious processors. And this is also similar in spirit to Jean-Pierre Changeux and Stanislas Dehaene's global neuronal space that they proposed in 1978, which is here, and which receives and outputs information about the past, the present, the future, the value. You can evaluate events, and you can also produce actions. Okay. Now, mathematics and consciousness. This is very, very, very weak, but it's an attempt. It's not original. You can think of a graph G with vertices and edges. Each vertex would represent some brain areas, visual, auditory, somatosensory, and so on and so forth. The mathematical description of this vertex is a set of possibly stochastic neural field equations. Each edge E represents the connections between the various brain areas. The connections can be anatomical through the so-called connectome, collected using diffusion MR, or functional. And this connectivity, these edges may introduce delays. So you have to work about delayed stochastic neural field equations. I didn't have time, but it's a very active research area, delayed neural field equations. And the mathematical changes for you folks are to provide mathematical models of the four signatures of consciousness I just described, such as bifurcations, metastable states, like Effa was talking about yesterday, and so on and so forth. mathematical challenge is to study rigorously how and when these can appear in the solutions of the neural field equations and provide mathematical models of the global neuronal workspace. This is an agenda that is going to last for at least a century. Other challenges, computational, already going on, Human Brain Project, The Virtual Brain from Jerza et al., or clinical to study consciousness in people in coma, David Nakash and Stanislas Dehaene. Good conclusion. Very short conclusion. You've all heard about the unreasonable effectiveness of mathematics in physics, put forward by Eugene Wigner in 1960. And to me, you may disagree, that this effectiveness is not yet, hopefully, verified in neuroscience or in biology in general. And one reason for this may be that there are few of the mathematical problems, and some of them are very difficult, raised by these disciplines, neuroscience and biology, that are studied by professional mathematicians. And one of the goals of our community is to bring professional mathematicians to work on, talk them into working on neuroscience problems. This is the end of my presentation. And as I told Eva and the members of the committee, I would like to announce a new journal, M&A, for mathematical neuroscience and applications, which is a bifurcation from the Journal of Mathematical Neuroscience published by Springer, which ends publication this year. Okay, it focuses on mathematics for neuroscience. You can read this here. The most important part, hopefully, is this one: is that it follows the Diamond open access model, which means it's open access, but it's free to the readers and free to the authors. It's free to everyone. This is science for everyone, and this is thanks to the support of epsciences.org, which is a non-profit organization. And this is a screenshot of the webpage that you can go to, mna.epsciences.org. And we are looking forward to receiving your submissions. You can also look at the set of members of the editorial boards. Many of them are on this committee for ICMNS. I thank you very much for your attention. I may answer some questions. I try to stop my clock here, which I followed carefully.
Thank you very much, Olivier, for this beautiful talk. So I forgot actually to announce to everybody that questions, of course, you can ask them like yesterday by just chatting to us, or you can also raise your hand. So downstairs on the Zoom block, there is the possibility to raise your hand if you have any question, and I can unmute you such that you can address directly to Olivier if you wish to do so. So let me just quickly check if there what I should say. So for the moment, I don't see. So maybe I start with the question. So you have been speaking a lot about neural field models all over your talk, also about stochastic ones. Did you consider particular models for stochastic models for these connectivity matrices? And if yes, what would be relevant ones according to you?
In the analysis, I presented the analysis that James Inglis and I did. The function J, the connectivity function, is non-random, is deterministic.
Yeah.
It would be a nice follow-up to our work to suppose that it's stochastic. And I think it would be a good idea. It's more difficult, but it's probably feasible to make some assumptions on J of X, Y, and T as a random process. But we haven't done so. But it's certainly an interesting, valuable research area. Thank you for the question.
So I see that
Well, the talk was difficult.
Huh?
The talk was probably a bit difficult and fast.
It was fast, of course, because you gave an overview of 20 years of research, or maybe even 30, I don't know. Yeah. So you were also speaking about these, so somehow these long-range interactions in part 2. So it's like a phase transition. So if you add a little bit of them, it completely changes the picture. So
yes.
And if even more increase this small parameter epsilon, is something else happening?
This small parameter epsilon is something which is biologically verified. From what I've read from the literature, it is known that the long-range connections are weaker than the short-range connections, which allowed us to consider the solutions as perturbed solutions to the local homogeneous solutions with the D4 symmetry group. What's important in this part is the role played by group theory. Of course, I didn't have time to go into the very interesting details which can be found in our JMN paper. But I really think that for many sensitivity procedures of the human mind, this group theory approach is important, but also for other things. As I stated at the end, I think group theory is underused in mathematical neuroscience, and I would like to push young people into using group theory in the mathematical modeling of the human brain. It would be also very exciting to combine this with the stochastic aspects, but it's even harder. So
yes.
Yeah.
There is a question by Abed Make, but I'm not sure that I understand it. So here's the question. Can you give us your opinion on using information theory to try understand the fingerprints of what? Of connectivity?
To consciousness.
Consciousness, yeah, I just didn't have the time to.
Sorry. So information theory to understand consciousness?
Yeah, basically there's this attempt in the field to use information theory to pursue consciousness. Do you think that what you have presented so far, these 4 fingerprints, can we also try to approach, like try to understand them or approach them from the direction of information theory?
Yes, I think so. I haven't talked about information theory in my talk. I couldn't talk about everything, but it is certainly something very worthwhile looking at. Of course, this is connected to notions such as entropy, mutual information, and so on and so forth, which are really natural to use in the area of stochastic neural field equations. So there's and also PDEs, of course, but this has not been done. This is certainly an interesting approach, and I would encourage you. All approaches. This is such a difficult and outstanding problem that all mathematical approaches that seem to be relevant should be tried, and probably the answer, if an answer ever comes out, the answer will require a large combination of different mathematical approaches. What's very nice, what's very nice is that Dehaene, in particular, Dehaene and his group of people have put this, have made a part of consciousness, this ability to report what you feel, what you see, amenable to experiments, well-conducted experiments, well-understood experiments, and therefore to modeling, because we can now confront our models to experimental results.
Thank you, Olivier. Maybe we have time for one more question. There's one by Romain. So I will read it if Romain doesn't want to ask himself. So brain is characterized by its learning capacities. How would you see synaptic plasticity fitting in the research axis that you described?
Oh, very simply, very simply. The most obvious one is to put some dynamics on the connectivity function J. So that would allow you to incorporate some aspects of learning at this resolution level. So add one more equation to the neurofield equations, namely one equation or maybe several equations on the J function. And you can probably account for some plasticity phenomena again at this very coarse level. Remember, neural mass is, I haven't said it very clearly, but a neural mass is an average of activities over a small volume in the brain. So it's a sort of a mean field approach. So you're already neglecting the activity, you're neglecting the activity of individual neurons. But that may be okay for applications such as consciousness or learning, learning.
Mm-hmm. Thank you very much, Olivier. I think we have to stop here to start your